I'd like to thank everyone for joining me to a beginner's guide to CHIP. My name is David Grotsky. I'm the Epigenetics specialist in scientific support at Abcam for the Americas. I'd now like to start by introducing you to CHIP. Chromatin immunoprecipitation, or CHIP, is the selective enrichment of a chromatin fraction containing a specific antigen. This antigen is a specific protein of interest, and you enrich for this protein bound to DNA by pulling it down using an antibody. CHIP allows you to study gene regulation by determining the localization of a protein of interest, for instance a transcription factor, in the genome at all of its binding sites and under specific conditions, such as different disease states or treatments. You can also compare enrichments of a protein or protein modification, such as a histone modification, at different loci. You can map the location of a protein of interest across an entire locus, and you can quantify a protein or protein modification at an inducible gene over a time course. Of course, for CHIP, the starting template is chromatin, which is a protein and DNA complex whose basic function is higher order packaging of DNA into the nucleus. If all 3.2 billion base pairs of DNA in the human genome were unraveled, it would stretch to around 5 feet long. So chromatin allows all of the DNA to fit inside the nucleus. Chromatin is also extremely important in regulation of gene transcription. The most basic unit of chromatin is the nucleosome, which consists of DNA wound around a core of 8 histone proteins. The nucleosome core consists of approximately 147 base pairs of DNA wrapped around the octamer, made up of 2 copies each of histones H2A, H2B, H3, and H4. Histone H1 sits on top of this structure to keep the wrapped DNA in place. More relaxed chromatin is termed euchromatin, which tends to be gene-rich due to its open and accessible structure. As chromatin condenses further and further, it becomes what is called heterochromatin, which contains repressed and silent loci, as well as repetitive elements. Finally, there is even higher level packaging to form metaphase chromosomes during mitosis. Here are classic electron micrographs showing what chromatin looks like, with the distinctive beads on a string structure on the left and more compacted chromatin on the right. Now here's a closer up view of the nucleosome core with the 8 histone molecules and DNA tightly wrapped around it. The diameter of a nucleosome is around 10 nanometers, and histones are unique in that they have N-terminal tails that stick out from the nucleosome core, whose residues are targeted for modifications that play different roles in the regulation of chromatin structure and therefore affect gene transcription. Histone modifications, along with DNA methylation, are the main epigenetic mechanisms that control gene transcription. When a gene is switched on and transcription is possible, the chromatin is open and relaxed so that transcription factors and other binding elements can bind freely to promoters. When a gene is switched off and transcription is impeded, the chromatin structure is condensed so that these factors can't bind to the DNA. Here is a diagram of some of the different modifications that occur on the histone tails. Histones H3 and H4 are the most commonly modified histones whose residues are targeted for acetylation, methylation, phosphorylation, deamination, and ubiquitination. Acetylation is generally associated with gene activation and occurs mostly at promoter regions, while different and specific methylation modifications are involved with either gene activation or repression by relaxing or condensing chromatin. For example, di- and trimethylation of H3K4 and monomethylation of H3K9 are associated with activation, whereas di- and trimethylation of H3K27 and trimethylation of H3K9 are associated with repression. Deamination occurs when an arginine residue is converted to a citrulline residue. Phosphorylation has roles in DNA repair, transcriptional regulation, and chromosome condensation, and ubiquitination appears to play a role in DNA repair. I'd like to focus a bit more on acetylation and methylation, as these modifications are most commonly studied by Chip. Acetylation occurs on lysine residues, and there are specific enzymes that add or remove acetyl groups to lysines, histone acetyltransferases and histone deacetylases, respectively. On the right, you can see a representative Chip experiment using an H3K27 acetyl antibody. This antibody specifically recognizes histone H3 when it's acetylated at lysine 27. Here, we pulled down DNA that was bound to this histone modification and performed qPCR using primers for several different loci to determine where the modification is located. You can see that this modification is located at promoters of genes that are constitutively active, such as GAPDH, RPL30, and ALDO-A, which we expect since acetylation is associated with gene activation. We can also see that this modification is not present at inactive genes, which we also expect. Similarly, for methylation, there are enzymes that add or remove methyl groups to lysines and arginines, histone methyltransferases, and histone demethylases. On the right, I'm showing a Chip experiment using an antibody specific for histone H3 when it's trimethylated at lysine 9. We find this modification present at inactive genes such as MyoD and Serpina, as well as at heterochromatin, while you can see that it's mostly absent from active loci. Of course, Chip isn't only for histone proteins. You can also use Chip to analyze the location of transcription factors or other DNA-binding proteins. Here's a cartoon of RNA polymerase II, which sits at the promoter along with transcription factors and moves along the DNA as transcription progresses. If you perform Chip to see where RNA polymerase II binds, you can see strong binding at active promoters, such as those of GAPDH and gamma-actin, and you can see the polymerase along the entire gamma-actin gene as transcription progresses. As expected, RNA polymerase is not detected at inactive genes. Now I'd like to go over the Chip protocol in depth. Here's an overview of the Chip protocol. There are two main ways you can do Chip. Cross-linking Chip, which can be used for any protein but must be used for proteins that weakly bind to DNA, and native Chip, which can be performed when analyzing histones. I'll go into greater detail about the two a bit later. In cross-linking Chip, DNA is cross-linked to proteins, typically using formaldehyde. Cells are lysed, and the DNA is then fragmented by sonication into random fragments of 200 to 1,000 base pairs. In native Chip, the DNA and proteins are not cross-linked, and micrococcal nuclease is used to digest the DNA, which can produce DNA fragments at the level of a single nucleosome, or around 150 to 175 base pairs. It is incredibly important to start with the best chromatin template as possible, so the first two steps are probably the most important and must be optimized for each experiment. Next, a specific antibody of interest is added to bind the protein which is bound to DNA, and this complex is then immunoprecipitated, pulling down only DNA fragments that are directly bound to the target protein. Crosslinks are then reversed if you're doing cross-linking Chip, and the DNA can be purified for later analysis by PCR, hybridization to a microarray, or sequencing. The abundance of DNA pulled down can be compared to the input, or starting chromatin amount. The first step in cross-linking Chip is to cross-link the DNA to proteins. Most commonly, this is done with formaldehyde, which cross-links protein to DNA, RNA, and other proteins. Less commonly, chemicals such as cisplatin can be used, and cisplatin cross-links proteins to DNA only. Ultraviolet light is also a cross-linker, but is not used in Chip, only in RNA immunoprecipitation or cross-linking immunoprecipitation to analyze protein interactions with RNA. Cross-linking fixes molecular interactions and freezes them at a given point in time. Importantly, formaldehyde creates reversible crosslinks, and typically you'll use a concentration of 0.75 to 1.5% for 2 to 30 minutes. It's important to perform a time course to see what cross-linking time gives you the best results. It's important to keep in mind that if you cross-link for too long, This can lead to the reduction of antigen accessibility, altered epitopes, and a decrease in protein binding, whereas if you don't cross-link enough, the proteins will dissociate from the DNA. Next, whether you're doing cross-linking chip or native chip, you need to fragment the DNA into smaller sizes. DNA fragmentation is important because it makes the chromatin soluble, and the DNA fragment size determines the mapping of the target antigen. If your fragment size is too large, you could be pulling down a protein that's far away from your actual DNA region of interest. So this step requires optimization. You should try different sonication times for cross-linking chip and different concentrations of micrococcal nuclease for native chip. After sonication or enzymatic digestion, you should remove some chromatin, reverse the crosslinks, and purify the DNA to run in a gel alongside a DNA ladder as shown in this figure to determine what condition gives you the most appropriate sized fragments. You're ideally looking for fragments of 200 to 1,000 base pairs by sonication or around 175 base pairs by enzyme. Keep in mind the efficiency of sonication is affected by foam, cell density, the cross-linking time, and the cell type you're working with. The next step is the immunoprecipitation. This is where you add a specific antibody to bind the protein bound to DNA and then pull it down. The amount of antibody needs to be optimized, and chip typically requires a large amount of antibody, anywhere from 1 to 10 micrograms. Next, you add protein A, G, or a combination of protein A and G beads that will bind to the antibody. These can either be agarose, which you'd centrifuge, or magnetic, which would be pulled down in a magnetic tube rack. Next, there are several wash steps to remove nonspecific binding. Finally, you need to recover the DNA that was pulled down as part of the antibody-protein complex. First, you elute the antibody from the beads using SDS and heat. Then you reverse the crosslinks if you're performing cross-linking chip. This is done with salt and heat. Next, the proteins in RNA get degraded using proteinase K and RNase A. And finally, you'll purify the DNA using a phenylchloroform extraction or a PCR purification kit. Be sure to include your input sample in all of these DNA recovery steps. Next comes the downstream analysis to determine what sequences of DNA got pulled down. This is done with qPCR, chip-on-chip, or Chip-seq. I've already showed some examples of qPCR data. Keep in mind that when you use qPCR, you need to have target loci in mind, and you need to design primers for the amplification. Chip-on-chip and Chip-seq allow you to perform chip on a genome-wide scale. In chip-on-chip, the chip procedure is carried out as it normally would be until after the DNA is purified. At this point, the immunoprecipitated DNA is amplified and labeled fluorescently. The input DNA is also amplified and labeled with a complementary fluorescent probe, and both sets of labeled DNA are combined and hybridized to microarrays corresponding to specific domains, loci of interest, or the whole genome. The ratio of signal from the fluorescent labels of the chip DNA and control DNA allows enriched regions to be identified. There are some limitations of chip-on-chip, though as technology advances, these are becoming less of an issue. The first is that a large number of cells are required for chip-on-chip experiments. Chip-on-chip is also not sensitive to all of the repetitive elements in the human genome. A large number of arrays are necessary to cover the entire genome, and finally, chip-on-chip could be susceptible to amplification bias after the DNA amplification. More commonly, Chip-seq is used to analyze protein-DNA binding interactions on a genome-wide scale. In Chip-seq, the pull-down DNA sequences are purified and amplified, followed by high-throughput sequencing of the fragments. These sequenced fragments then get mapped and aligned to the genome, and if there are many sequenced fragments that overlap at a specific spot, they form a peak. These peaks indicate that this sequence was bound to the protein that got pulled down by the antibody. Here's an example of what the peaks look like in these sequencing tracks from a publication that was looking at histone modifications genome-wide. Here they're showing results from a region with two active genes, STAT1 and STAT4, and results from a region containing an inactive gene, MYO1B. You can see that activating histone marks, such as H3K4 methylation at the top left, are present at the active genes, especially at the gene promoters, but are absent from the repressed gene. Similarly, if you look at marks of repression, such as H3K27 and H3K9 di- and trimethylation, you can see that these modifications are found along the inactive MYO1B gene. To show how powerful CHIP is, I'd like to show this figure from the same publication. Here the authors created a high-resolution map of histone modifications on a genome-wide scale. They correlated histone modifications and several other proteins, such as RNA polymerase II, in around 13,000 genes in resting human CD4-positive T cells whose expression levels are known. They separated the genes into groups based on their expression levels, either high, medium, low, or silent genes, as indicated by the different colors, and aligned them relative to the transcription start sites. We can see the binding of RNA polymerase II right at the transcription start site. We can see the binding of RNA polymerase II right at the transcription start site of the active genes, and if we look at the histone modifications, we can see a nucleosome-depleted region right at the transcription start site where no histones are bound, with the marks of activation, H3K4 methylation, present around the start site in active genes, and the mark of repression, H3K27 trimethyl, present at the silent genes. All of this CHIP data has allowed us to see exactly which and where histone modifications are present along an entire active gene, as well as in inactive genes, which I'm not showing here. When transcription is happening, histone modifiers introduce activating histone modifications as the polymerase travels along the DNA. The promoter region has a very well-defined chromatin signature. There's a clear nucleosome-depleted region where the polymerase binds, and large increases in activating H3K4 methylation and acetylation. As transcription continues along the gene, modifiers introduce new modifications to allow the polymerase to move along the DNA. I hope these few examples show how powerful and useful CHIP can be. Now I'd like to spend some time going over protocol optimization and troubleshooting. As I mentioned earlier, there is native CHIP and cross-linking CHIP, and each has its own advantages and disadvantages. With native CHIP, you typically get more efficient precipitation of the DNA and protein, as well as greater specificity because the binding is more predictable. You also have higher resolution, as low as a single nucleosome, compared to 200 to 1,000 base pair fragments you get with sonication. The disadvantages of native CHIP include the fact that it's only for histone proteins because the DNA is tightly interacting with these proteins. If you want to do CHIP of transcription factors, since their binding to DNA is not strong, you'll have to cross-link to ensure the protein-DNA interaction remains intact. Another disadvantage is that since the nuclease digestion is selective, the digestion may bias input chromatin, and there are also chances of chromatin rearrangement. And of course, if you use too much nuclease, you could over-digest the chromatin. Cross-linking CHIP, on the other hand, is much more flexible and can be performed with histone and non-histone proteins on all cell types, tissues, and organisms. In addition to DNA protein analysis, you can also analyze RNA protein and higher-order protein-protein DNA interactions, and there is a reduced chance of chromatin rearrangements. There are, of course, several disadvantages of cross-linking CHIP, including the fact that overfixation can prevent effective sonication, enzymatic digestion won't be possible after formaldehyde treatment, formaldehyde can alter the binding of the antigen, and it has a lower resolution than native CHIP, since you can't use micrococcal nuclease for digestion. Hopefully, this will help determine if native or cross-linking CHIP is more appropriate for your experiment. I'll start with optimizing your protocol by going over how to choose the right antibody. Most antibodies will not work for CHIP, so a good place to start is to look for antibodies that have been fully characterized for CHIP. You should also look for specificity testing, such as peptide array data, Here's that H3K4 trimethyl antibody I showed earlier, and we can see the antibody is binding strongly only to the K4 trimethyl peptide, with minimal or no binding to these other peptides tested in the array, indicating that the antibody is specific for the trimethyl K4 modification. We're developing new, highly specific rabbit monoclonal antibodies that are all specificity tested by a new peptide array whose data is available on every datasheet. This peptide array contains 501 different modified and unmodified peptides, and each peptide is printed on the array at 6 concentrations. The circles represent the affinity between the antibody and a peptide. Red circles show binding to the antigen-containing peptides, while non-antigen-containing peptides are in blue. You can click on a link for more data showing what each peptide is, and all of the batches of our CHIP antibodies are tested by peptide array. If an antibody is not tested for CHIP, but you're interested in trying it out, look for antibodies that have been tested for immunoprecipitation, immunohistochemistry, or immunocytochemistry. These applications recognize the protein's native conformation, similarly to CHIP, rather than a Western blot-only antibody that might only recognize a protein in its denatured form. If you'd like to test one of Abcam's antibodies in CHIP, you can use our AbTrial testing program, which I'll discuss more later. Finally, with regards to clonality, polyclonal and monoclonal antibodies both have advantages and disadvantages. Polyclonal antibodies recognize several epitopes, which is great for cross-linking CHIP, where epitopes might become masked, but they do have some batch-to-batch variation. Monoclonal antibodies recognize only a single epitope, so if the epitope is masked, the antibody won't bind, but there is very minimal batch-to-batch variation. Look out for our new rabbit monoclonal antibodies for CHIP. As I mentioned earlier, probably the most important step in optimizing your CHIP protocol is beginning with a really good chromatin template and the correct fragment size. For cross-linking CHIP, this involves finding the appropriate cross-linking and shearing time, and for native CHIP, this involves finding the appropriate amount of enzyme and digestion time. You should perform a time course to determine the right amount of time to cross-link, which could be anywhere from 2 to 30 minutes, but 10 to 15 minutes is usually sufficient. If you're looking at protein-DNA interactions that are mediated by other proteins and are not directly bound to DNA, you may need to try a dual cross-linking strategy, where you cross-link with a combination of formaldehyde as well as an additional cross-linking chemical such as EGS or DSG. Getting the right fragment size is of utmost importance. You must try different sonication times and then purify DNA to run in a gel to figure out what sonication time will get you the most appropriate size. You typically want fragment sizes of 200 to 1,000 base pairs. For native CHIP, you can get fragments of around 175 base pairs. Next, you should determine the proper amount of antibody to use for your CHIP. Typically, around 4 micrograms of antibody per 25 micrograms of chromatin is appropriate, but the antibody should be titered to improve the signal-to-noise ratio. The composition of your wash buffers is important. You should determine the appropriate amount of sodium and lithium chloride to provide the appropriate stringency. This could be from 250 to 500 millimolar. Having a wash buffer that isn't stringent enough will lead to high background, while a buffer that is too harsh will destroy specific antibody interactions. Since CHIP is difficult and time-consuming to perform, it's very important to have the right controls to determine if the experiment worked properly. These include a negative IP control, which would be an isotype IgG control or beads only, positive and negative control loci for qPCR, which would be a region or gene where you know a protein should be bound and shouldn't be bound, and a non-template control for qPCR to make sure there's no contamination in your PCR. Positive control antibodies are extremely important. If you're looking at an active locus, you can use antibodies that recognize H3K4 trimethyl or H3K9 acetyl, for instance. And if you're looking at a repressed locus, you can use antibodies that recognize H3K9 trimethyl or H3K27 trimethyl. You can also use a histone H3 antibody, which should be present everywhere. Here you can see the presence of H3K4 trimethyl at active loci and H3K9 trimethyl at heterochromatin. Now on to troubleshooting. What if you have high background in your nonspecific antibody control? So this would be either your isotype IgG control or beads-only tubes. There could be nonspecific binding to the beads, so I always recommend including a pre-clearing step. You could have contaminated wash buffers, so it's good practice to always use fresh buffers or replace your buffers if you're getting high background. And some beads inherently have high background, so it's good to try different beads and different blocking strategies to see which provide the lowest background in the nonspecific control. What if you have low resolution with high background across large regions? This is most likely due to the DNA fragment size being too large. Fragments should definitely not be larger than 1.5 kilobases, and ideally should be between 200 to 1,000 base pairs. And keep in mind, if you're able to do native chip, you can get the best resolution of a single nucleosome. What if you're getting low or no signal? Well, lots of things could be wrong. The DNA fragment size could be too small, so make sure you run your fragments on a gel to see if they're the correct size. If you're performing cross-linking chip, the cells may have been cross-linked for too long, which can reduce the availability of epitopes, so be sure to perform a time course and quench with glycine. It could be that you don't have enough starting material. Chip generally requires a large starting input, around 25 micrograms of chromatin per IP, or 3 to 4 million cells per IP. You could also not have used enough antibody. 3 to 5 micrograms is usually sufficient, but up to 10 micrograms might be required if there's low or no signal. You could be eliminating the specific antibody binding if your wash buffers are too stringent, so don't use more than 500 millimolar of sodium or lithium chloride. Make sure your cells are sufficiently lysed. Something like a RIPA buffer should work well. Keep in mind that your result might be real if there's no antibody enrichment at the region of interest, so be sure to include a positive control antibody and locus to ensure the chip is working. Make sure you know when to use native versus cross-linking chip. Cross-linking chip is more suitable when analyzing proteins that have weaker DNA affinity, so cross-linking will be required to keep proteins associated with DNA, and native chip can really only be performed if you're looking at histones. If you're using a monoclonal antibody, it may not be suitable for cross-linking chip due to epitope masking, so try an antibody that's tested for chip or a polyclonal antibody. And make sure you're using the correct affinity beads, so make sure the antibody species and immunoglobulin bind to the chosen beads, or use a protein AG mix. What about problems with the PCR amplification? If you're getting high signal in all of your samples after PCR, including the non-template control, your PCR solutions may be contaminated, so you should use new solutions. And if you're getting no amplification in your samples, be sure to include a standard or input DNA to confirm the primers are working. If you're performing or planning to do Chip-seq and need help with the analysis, we have a step-by-step guide to Chip-seq data analysis webinar available to watch at any time on our website. You can find it under Scientific Webinars in the Events section of our homepage, or by navigating to the website listed here. I'd like to turn now to the resources Abcam provides to help scientists with their research. I mentioned the AbTrial program earlier. This is a program where you can use our products in an untested application or species without financial risk. This is a great program if you'd like to try out one of our untested antibodies in chip. If you send us your results in an Abreview, whether they're positive or negative, we'll send you a discount code for the full value of the product that can be used on a future order. If you'd like to enroll in this program, you can visit abcam.com/abtrial or contact our scientific support department. We also have our Abreviews program. These are user reviews from customers like yourselves that show whether or not a product works and exactly what they did to get the product to work. You can submit a review for any of our products on a scale of 1 to 5 stars, and we publish all of them, good and bad. Here you can see this H3K9 acetyl antibody has 32 reviews, and if you click on the circled link, You're able to filter the reviews by application, species, and by rating, and you can see the specific protocol information and an image for each review. We also gather references for all of our products. When we find them or hear about them from customers, we make them easy to access on every datasheet by clicking the circled link. You can see this antibody has 88 references, and when you click the link, it gives you a list of the publications with the convenient link directly to the PubMed entry, as well as the application and species that the antibody was used in. Abcam also has many products for your epigenetics research. We're knockout validating our antibodies, so you can be sure that these antibodies are specific. We're using CRISPR-Cas9 technology to completely knock out the gene in haploid cell lines. These antibodies are easy to find on our website. You can just search for knockout. You can filter for knockout validated antibodies on the left side of the search page if you're looking for a specific target. And all of these antibodies have this KO validated logo. We're showing the knockout data on all of the antibody datasheets. In this Western blot for AKT1, you can see in the right panel the wild-type sample has a band at the expected molecular weight, and the band is gone in the knockout samples. Beta-actin is used as a loading control. We're also performing immunocytochemistry. As you can see here, with this Ki67 antibody, the signal is gone in the knockout samples in the lower right-hand quadrant. We also have over 200 kits for studying different epigenetic mechanisms. For sample preparation, we offer chromatin, histone, and nuclear extraction kits, as well as DNA extraction kits for whole cell, genomic DNA, formalin-fixed paraffin-embedded DNA, urine DNA, and plasma and serum DNA. If you're studying DNA methylation, we have kits for bisulfite conversion, bisulfite sequencing, 5-MC and 5-HMC quantification, methylated DNA preparation, and methylated DNA immunoprecipitation, and hydroxymethylated DNA immunoprecipitation. For RNA methylation, we have RNA bisulfite conversion kits and M6A methylation quantification kits. For chromatin analysis, we offer Chip and Chip-seq kits, as well as kits to analyze histone modifications, such as HDAC and sirtuin assays, acetylation, methylation and demethylation quantification kits, and kits to measure histone phosphorylation. If you're studying microRNAs, we have our Firefly multiplex microRNA profiling platform, which you can find a ton of information about on our website. Finally, we are here to help. If you ever have questions or problems with our products, don't hesitate to contact us by email or phone. We offer support around the globe and in many different languages. Also keep in mind that all of our products are covered by our AbPromise guarantee. If you're using a product in a tested application and species and it's not working as it says it should on the datasheet, just let us know and we'll help you get the product working or give you a replacement or refund. To contact us, just click the Contact Us link at the top of our website or email us here. I'd like to thank everyone for watching.